The key feature of organisms on which evolution concentrates is reproductive success. And reproduction, being the sort of centerpiece of the life of an organism, has evolved in ways that are especially interesting and that have many implications for medicine. We're going to start our discussion of that in this section with the origin of mammalian reproduction. And this is a case where conditions that are now important medically go back about 190 million years. The whole eutherian reproductive system is an evolutionary innovation. By eutherian, we mean those mammals that are not kangaroos or possums or things like that. They are mammals that have live birth with not only a live birth with a placenta, which to a certain degree is primitively developed in kangaroos, but they have a long period of pregnancy. The reproductive system in females is derived from ancestral organs that produced eggs that came out through a cloaca. The innovations in mammals include the uterus, the endometrium, the placenta, and the vagina. Those are all new structures. Those are all things that evolved about 190, 150 million years ago. Those innovations are accomplished by changes in the developmental control of cells and tissues. And in this case, the genes that are used to control these developmental fates had previously evolved to control the development of the body axis and the limb. And as we'll see, there was a very interesting thing happen that allowed the expression of these genes to switch and to start to control new structures. Now, I'm going to rehearse some of the general features of developmental genetics in order to provide background on what's happened in the female reproductive tract. The key issue in development is how to coordinate the fate of cell types. The fact that organisms consist of cells and that we really need to worry about how cells interact with each other, we owe to Schwann and Schleiden back in 1839. Cells communicate with each other. They send and receive information on where they are. We call that positional information. That would be top, bottom, front, back, left, right, that sort of thing. They use information to change the fate of cells, so cells then know that they're going to develop based on where they find themselves. And it helps to remember that every cell in the body, in our case the exception is red blood cells, contain all the information necessary to produce the entire organism. So the difference between a liver cell and a heart cell isn't that the liver cell has lost the genes that would have made it into a heart cell. It's just turned them off and it's turned the liver genes on. It has all the genes. That means that development is controlled by regulating gene expression, not by discarding genes. Much of trait evolution has changed when and where genes are expressed. It hasn't changed the DNA sequences of the structural genes. In fact, one of the particular games that developmental biologists like to play is trying to reimagine the history of life, seeing how many of the different kinds of plants and animals they might be able to get just by changing the developmental control genes and not by having any mutations in the structural genes at all. That's set off of a bit of an interesting controversy. In fact, both of those things go on, but there's no doubt that in terms of development, the control genes are important. So what is development doing in evolution? Well, it shapes the variation that's presented to selection. What do we mean by that? Well, probably one of the easiest examples would be there is no way that just regular mutation is going to produce mutations in my offspring that have 6 legs and 12 arms. They're going to have 2 legs and 2 arms. The arms might be a little bit bigger, a little bit smaller, that kind of thing, but there are going to be 2 of them. So that's what I mean by development shaping the variation. There are only a limited number of variants that can be produced. Developmental mechanisms that are shared among related organisms constrain responses to selection in ways that are shared by relatives. So, for example, all insects share related control mechanisms in development. All mammals share related control mechanisms in development. And that means that selection is going to operate on variation in insects in one way and on variation in mammals in another. So that's why species resemble shared ancestors in basic body plans, and that's why ancestry influences responses to selection. Those influences can be very strong. Another important aspect of development is that genes can only build organisms out of materials and complex systems of chemistry and biological interaction. They don't have complete freedom there. They can then only work with the properties of those materials. They can't directly control every detail of the phenotype. Now, in these two statements, what I'm really saying is the metaphor that the genotype is a blueprint for the production of the organism is actually misleading, because a lot of the information on how to build the organism is in the materials. It's not in the DNA sequence. If a DNA sequence you think of as a metaphor tries to select a material to build bones in the vertebrate body, and it hits upon hydroxyapatite, it is getting a lot of information for free. It is getting the information that if you put stress on that material, it will strengthen in the direction of the stress. A very convenient property for a bone, but not something that's in the DNA sequence. That's just given for free by hydroxyapatite. Then the final point here on what evolution does in evolution, what development does in evolution, is that it generates the properties of developmental systems by modifying existing systems. It just works with whatever variants are available at the time. It doesn't design organisms a priori the way an engineer would. And that's why when we look into developmental systems, we find little puzzling things in them that are relics of history and not design properties of ideal systems. The major players in multicellular eukaryotic gene regulation are sketched here. So what you have here is what looks like a paper clip is actually a bend of DNA. And sitting down on that DNA are a group of little colored blobs here, which are transcription factors and enhancer systems, sequences, and when they are correctly arranged on a piece of DNA, then RNA polymerase will sit down, and it will sit down in the promoter region and start to begin transcription in this direction. As you can see, there are quite a few elements here that have to come together. Each one of these is a gene product, which means that there are many different combinations of transcription factors that could be out there, and that's one of the things that gives them specificity for this particular gene. You have to have a combination of a number of things all sitting down in the right place to get this gene to turn on. How does that start off? You have a simple egg, it grows up into a complex organism. It starts off with a concentration gradient that gives some positional information. So we know, for example, in the fruit fly egg that this is laid down in the mother's body. It is molecules that she puts into the egg that determine by their concentration what is the front, what is the back, and so forth. So that's body axis. That works in the limb as well, where we have proximal, distal, and then we also have posterior, anterior, posterior, and dorsoventral for the body axis. So those big sort of symmetrical structuring relationships, those symmetries, are laid down initially by concentration gradients. Then, when cells start to develop, and they know from the concentration gradients what kinds of transcription factors they should produce, those then signal specific areas where only a subset of genes is expressed. That's how you can get heart in one place and liver in another. That gene expression, as we've seen from the preceding slide, is regulated by combinations of activators and repressors. Combinatorial control allows a huge diversity of cell-specific gene expressions. There are hundreds of different kinds of cells in the body, so each one of those is elicited by different combinations of transcription factors. The genes that produce transcription factors are regulated by genes that produce transcription factors. That builds the possibility of genetic signaling networks. And the same genes that play one role in the embryo can play another role in the adult. We're going to see that both in terms of the limb, the way the vertebrate limb is controlled developmentally, and in terms of the female reproductive tract. In both cases, the genes that are controlling that development were doing another thing very early in development. Generally speaking, the genes that control the general body plan switch on first, and then genes controlling progressive layers of detail switch on later. That's logical. And at the heart of a lot of this are developmental control genes. Now, developmental control genes, or genes that produce transcription factors, come in many different flavors. One family of such genes is very well investigated and is quite important in a number of areas, in body axis, in limb development, and in the female reproductive tract. That is the Hox or the homeobox genes. They are deeply conserved in their sequence and in their function, and they are collinear. So let me unpack that a little bit. They are called box, homeobox, from looking at the sequence of nucleotides in a DNA and seeing that there was a particular sequence that cropped up a lot. And all of these homeobox genes share that sequence. It's about 71 amino acids long, and it codes for a part of the transcription factor that will fold in such a way, it's helix turn helix, that it will precisely sit down into the DNA helix. So it's what allows them all to bind appropriately into the control region for gene expression. So that's where box comes from. It's a historical artifact of printing things out and then drawing boxes around all the DNA sequences that look the same and that were all shared by these genes. Homeo comes from homeotic. That refers to mutations in these genes that cause major changes in body plans. One of the first was bithorax. It was a mutation in Drosophila that caused Drosophila to have four wings, two pairs of wings, rather than just one pair of wings. And those are mutations that call forth the appropriate appendage in the inappropriate place. So that's the history of homeobox, and it's just shortened to Hox. Now, here is a rough phylogenetic tree showing at the top vertebrates, then here we have arthropods, down here we have annelids. And in all of these, you can find Hox genes, and they run on the chromosomes from 1 up to 13. And in all of these organisms, the genes that have lower numbers are controlling the development of the front of the organism, the ones that have intermediate numbers, the middle of the organism, and the ones that have high numbers, the end of the organism, the tail end. The other thing you can see is that in the vertebrate lineage, there have been 2 complete gene duplications, so that we have A, B, C, and D sets of Hox genes. And as you'll see in a few minutes, some of these sets have been used for controlling new structures. Collinear means what I've just explained in terms of the genes being on the chromosome in the same linear sequence as the parts of the body that they are associated with. If we look at that in the fruit fly and in the mouse, we can see that genes in the fruit fly that are Hox, would be the first member of the Hox family, are controlling mouth parts, and they are controlling hindbrain in the mouse, so they're up in the front part of the body. Genes a little bit further back are controlling the back of the head in the fly, and they are controlling the neck in the mouse. If we go a little bit further back into the middle of the body, We see genes that are controlling the thorax and the wings in the fly, and they are controlling the shoulder girdle and that part of the mouse. And if we go back further, we see that the genes that are at the tail end of the fly are also controlling things that are behind the heart in the mouse. The mouse has added further genetic control out into its tail. Now, that's remarkable because we can see the same linear sequence of control in organisms that last shared ancestors arguably about 600 million years ago. Now, I referred to the fact that in vertebrates, the whole Hox complex has been duplicated twice. One of the copies, the D copy, the fourth copy, is used to control the development of the forelimb. And if you look at the cells as they start to develop and you ask which genes are turned on, what you find out is that in the shoulder girdle, it's D9, Hox D9. Then if you turn on both D9 and D10, you get the upper arm. If you turn on 9, 10, and 11, you get the lower arm. You turn on D3, D12, you get the wrist, and you turn on D13, actually not D3. If you turn on D13, you get the fingers. So if you have all 5 on, you get the fingers. If you have 4 on, you get the wrist. If you have 3 on, you get the forearm. 2 on, you get the upper arm, and 1 on, you have the shoulder. Well, that's combinatorial control, okay? And it's done here with a duplicate set of Hox genes. Now, this is very interesting because this looks like you're just laying out a body axis. Instead of laying out a body axis, you're laying out a limb. And it's done the same way with the limb as it's done with the body axis. This whole set of developmental control switches was just plucked up and applied in a new place. Now, that's the background on developmental control genes. Let's take a look at the evolution of the female reproductive system. Here is a phylogeny, a phylogenetic tree, showing the relationships of amphibians, birds and reptiles, monotremes, that's the duck-billed platypus and the spiny echidna, marsupials, so that's possums and kangaroos, and placentals, that's the eutheria, okay? This split is about 360 million years ago, and this split is about 150 million years ago. This is about 170 million years ago, okay? So mammals started to really evolve and diverge from reptiles somewhere between 170 and 200 million years ago. The different parts of this are, these little blue circles are ovaries. This is the urinary bladder here, okay? And these are basically the uterus, and these are the kidneys. And this is the vagina, which is something new. This is a cloaca here, and the vagina is something new. The uterus is something new. Those are all elaborations of structures, tissues that didn't really exist as separate structures in the ancestors, okay? So as this process went on and mammalian reproduction evolved, at this point there was reduced reliance on yolk, even though monotremes still do give birth with eggs. Then in the marsupials, the cloaca divided, the oviducts differentiated more, the uterus started nourishing the embryo, and that's where the placenta originated. But in the placentals, the placenta really develops into what we now recognize as a full placenta. The embryo became invasive and there was longer internal development. What are the genes that are controlling that? It's our old friends, the Hox genes. This time it's the A set. So early in development, these genes were used to lay down the body axis, and then as the fetus develops, they control what becomes vagina, lower uterus, upper uterus, ovaries, and fallopian tubes. And look at the sequence. This is A13, this is 11, this is 10, and this is 9. So again, we have collinearity. That same battery of control mechanisms is being redeployed to help create a new structure, the mammalian female reproductive tract. Now, another thing which is going on here is that HOX A10 and A11, when the endometrium forms, downregulate the immune system in the endometrium, and that allows the foreign body, the fetus, or the conceptus, to implant. So they have a number of later functions as well. It's not only in setting up the development here, but much later in life during reproduction of adults, the A10 and the A11 gene jump in again, and they just take one specific part of the body and downregulate the immune system there to allow implantation to occur. A10 is also involved in endometrial maturation, as is A11, and A13 is also involved in umbilical arteries. Okay, so these are all things that they then do years later when the embryo is implanting. If we look across the ancestors, we can see the homologous organs. Okay, so we can use the expression of the control genes to show us what parts of salamanders, geese, duck-billed platypuses, and kangaroos are comparable evolutionarily to the female reproductive tract. You can see that HOX A9 is always being associated with oviducts. You can see that HOX A10 is being associated with structures that are then precursors of parts of the uterus. HOX A13 is always down at the bottom of the structure, being involved with the cloaca or with what becomes the vagina. So this is a method of looking back in history and then recognizing the homologous parts of the body. One of the innovations, very interestingly, was caused by the insertion of a transposon. So transposons are jumping genes. They are derived over a long period of time from retroviruses. And if you start where you have a number of genes that are not being expressed in a tissue, and a transposon that is carrying a regulatory element jumps in, then it will turn on all of those tissues. So we have an example here where a transposon that had a binding site for a transcription factor inserted above a target gene. Okay, so these are the target genes. And that brings that gene under control of that transposon. In the endometrial stromal cells, the transcription of prolactin, which is essential, is controlled by an enhancer that is derived from a jumping gene. So that's this right here. It was MER20 that jumped in, and that appears to have come into the mammalian genome at the time that placentation evolved. The point of this is that this gives a mechanism to understand how there can be an innovation in a tissue type. In order to get that, you have to change the developmental control of pre-existing tissues, and you can do that by having a transcription factor, which is being carried on a jumping gene, jump into and insert itself into a position that will then take control over the expression of a set of downstream genes. And it appears that this is precisely what happened at the origin of placentation. What it did was it rewired a big genetic network, okay? So if we look at the spot where that happened, it is a feature that's shared by armadillos and humans, but it's not shared with possums. And if we then look at the number of genes which are being controlled in a genetic network with armadillos and humans and possums, we get this Venn diagram. There are a lot of genes that are being controlled, okay? So if we look at the ones that are being expressed in all three, there are about 3,300. The ones that are uniquely human are about 200. The ones that are uniquely armadillo are about 1,300. That gives you some idea of the range of developmental control genes that are being well, the range of genes that are under the control of these transcription factor genes. Now, about 1,500 genes were recruited into the set of genes controlling endometrial development. 13% of these 1,500 genes are close to that transposon. And that transposon binds transcription factors that are essential for pregnancy, and it regulates gene expression in response to progesterone and to cyclic AMP. So we can see that there is at least correlative evidence that allows us to understand how this innovation, the endometrium, might have come about. And we even have some candidate mechanisms for understanding it. So to summarize, mammalian reproduction is an innovation, and it required the rewiring of a big genetic network. The mammalian reproductive tract develops under the control of the same genes that earlier laid down the body axis. Some of the innovation was made possible by the insertion of a transposable element carrying receptors for transcription factors. That is a very interesting picture of the way that innovations in general can be generated.